Hello. So in part one of this screencast, we started kind of picking away through the anatomy of the eye. We kind of briefly met the retina, which is this layer in the back of the eye. That's where the photoreceptors are. Those rods and cone cells who are responsible for converting the energy of a light wave into an action potential. We met the sclera, which is this outer white layer, the white of your eye, mostly providing structure and support. The cornea is a little window into that eye. We met the pupil, which is this hole that allows the light to continue to enter the eyeball. And then the iris that regulates the size of the opening of that hole. Then we met the lens, which helps focus the light, kind of bends the light to help it focus onto that retina. And then the ciliary bodies, which contain muscles that can change the shape of that lens. So we're just going to continue the journey through the eyeball here with the aqueous and vitreous humor. Both of these are fluids. The aqueous humor is the fluid in what's called the anterior or front part of the eyeball, and the vitreous humor is the fluid in the posterior or back portion of the eyeball. So they just kind of fill out the eyeball. And you might think, well, surely there's nothing interesting about a bunch of fluid floating around, Mr. Sanger, but oh, you're wrong. You ever heard of a floater? So sometimes maybe on a clear day, I usually see it on a sunny blue sky kind of day. I look up and I see these weird things in my eyeball and like I look to the right and they kind of drift to the right. I look to the left and they kind of drift to the left. And I'm like, is this a parasite? What is going on? Usually when I survey students, about half the class says, "Oh, I know what those are," and the other half is like, "What are you talking about?" So if you're in that latter group, sunny blue sky, look up and you might see these. All they are is little clumps of blood cells. And they just kind of find their way into the aqueous or vitreous humors. They get stuck in there, and then the light kind of passes through and gets distorted a little bit and creates this little weird image. They're harmless, so nothing to worry about, but that's what they are. Something that is something to worry about, though, is glaucoma. So glaucoma is a condition of the eye that is very problematic because what happens here is it's a drainage channel for the aqueous humor that gets damaged. So basically, here's kind of the yellow line represents sort of the production and drainage of aqueous humor. So your body is continually producing and draining, produce and drain this aqueous humor. Well, sometimes that drainage channel can be blocked. There was a Twins player, Kirby Puckett, who's quite famous, you may have heard of. And I think he got hit in the eye with a baseball, and that caused this to happen. And so if this gets damaged, all of a sudden the fluid is produced, but it's not draining. And so it builds up, builds up, builds up, and it kind of pushes back on this part of the eyeball. So all of a sudden there's this huge pressure in the back of the eye. And the problem is the retina begins to become damaged. The rod and cone cells, which are responsible for detecting the light wave and triggering action potential, they get damaged. And if they get damaged, they can't do their job, and you actually can go blind as a result of this. This is actually the reason why they recommend an eye exam every, I think, 5 years. I don't even know the number, but it's recommended that you see an eye doctor periodically. It's not so that they can see, can you read the eye chart, carefully. They're interested in that because you obviously want to get that corrected, and I think that's where a lot of the profit is in eye exams. Maybe I'm wrong, but really what they're looking for is to test for glaucoma. And there's two ways to test this. One is they have this kind of blue light, and they kind of move this thing closer and closer and closer and closer to your eyeball, and you're like, wow, that is really close to my eyeball. It's actually touching your eyeball, and it's measuring the pressure. Another way they can test for it is they have you put your little chin in a stirrup, and then they shoot a blob of air, a jet of air at your eyeball, and then they kind of let that air bounce off your eyeball and detect the velocity at which it ricochets. And if you have high pressure in your eye, then it's going to ricochet at a faster rate, and they can measure that and say, hey, you might have glaucoma, and then they can investigate further. So that's really the reason for an eye exam, is to make sure that you don't have glaucoma. Okay, that finally brings us to the retina. We've been talking about the retina this whole time, this layer in the back of the eyeball. This is where the photoreceptors are. So this is where the magic happens. Couple things about this. First of all, the center of the retina is called the fovea. That is where the photoreceptors are most dense. And pick an object in the room, just pick something, stare at it, and you'll notice when I stare at it, it's in focus. But everything in your peripheral vision is out of focus. It's a little bit blurry. And so, when you look right at something, the light waves are bouncing off that item and into your fovea, where the photoreceptors are dense, so you get a clear, crisp image. And everything in the periphery hits on the edges, which aren't as crisp. The other thing is the optic nerve. The optic nerve is what actually relays that signal to the brain. So you can see the retina is continuous with this optic nerve, and that is what is going to lead to the brain. Now, interestingly, there's no photoreceptors in that optic nerve, and so that actually results in a blind spot. And unfortunately, this isn't something that I can do virtually, but basically you can, if you draw a dot on a piece of paper, you can kind of close one eye, and it's complicated, but it's hard to describe virtually. But trust me, there's a blind spot. You can actually, if you position a paper just right, you can make a little black dot on the paper disappear, and that's because the light waves bouncing off that black dot are hitting right here where your optic nerve is, and you don't get that visual information. So that's kind of cool. Here's a video of laser eye surgery if you're interested in that. And again, it's a little traumatic, whenever you're cutting into an eyeball, that can be traumatizing, but if you're up for it, go ahead and pull up the slideshow and click that. What I want to do now, though, is get away from the anatomy of the eyeball as a whole and zoom in a little bit more on those photoreceptors, those rods and cones. Again, this region right here is the retina back here, this whole layer, and the rods and cones are in the very back of the eyeball, and they synapse onto these cells, which synapse onto the optic nerve and send the signal. So let's get to know them. The cones are cone-shaped, and they're sensitive to colored light. These are located mainly in the fovea, which is the center of the retina, and there are three types: red, blue, and green. These are actually the primary colors for light, meaning if you combine red, blue, and green in different ways, you can get all the other colors of the rainbow. When you're dealing with paint, the primary colors are red, blue, and yellow, but with light, it's different. It's actually red, blue, and green. So you have three types of cone cells sensitive to three main primary colors. We can actually play with our cone cells a little bit. This is kind of fun. Basically, just stare at that dot. Just stare at it for a while. You may want to pause the video in the middle so you can stare at it longer. I usually tell a joke or so to kind of delay. I'll tell you my joke. What do you call a cow with no legs? Ground beef. Okay, so we keep staring and staring and staring. Again, you might want to hit pause, but I'm just going to go ahead and click to the next slide. And if you keep staring, you should see a bluish dot. And the reason you're seeing that bluish dot, I'll go back to the red so you can stare at it longer. When you're looking at the red dot, what's happening is you're stimulating just one of those three types of cone cells. You're just stimulating the red. And so the red cone cells become fatigued and they don't function as efficiently. And so you can kind of picture if they're getting fatigued, when all three cone cells are equally stimulated, you get white. But if we kind of subtract these red ones that are fatigued, you're going to get the kind of bluish greenish. And so when we click to the plain white screen, since the red cone cells can't quite keep up, then you get kind of a bluish greenish circle. And maybe you can see that, maybe not. Again, if you pause it and do it for longer, it'll work. We can do the same thing with the blue. So here's my stalling joke. What do you call a cow with two legs? Lean beef. And then you're going to want to hit pause for this to really work. But now we're only stimulating the blue. So picture the red, green, and blue. If we're stimulating all three, but the blue can't quite keep up, then we're going to see a reddish greenish, which becomes orange. And so maybe you can see that after image. And then we can do the same thing with the green. And now we're going to see green gets subtracted, and so red and blue are going to make purple. But first, my joke to delay. What do you call a cow with one leg? Steak. So hopefully if you do this, you see kind of a purplish circle. So that's cone fatigue. Another interesting thing about photoreceptors is color blindness results when one or more of these different types of cone cells don't function properly. So red-green color blindness is the most common. And sometimes I'll ask volunteers, "Is anyone colorblind?" And if so, and the student is comfortable with it, I'll ask, "Can you just compare these two pictures and tell me what the difference is?" And a student who's colorblind has a really hard time telling the difference between these two sets of images. Here's kind of the typical palette of colors, and then here's the palette of colors that someone who's colorblind might experience. So they don't have those red cone cells functioning, and so that becomes kind of this greenish color. Note this actually came from a book about dogs' vision because most mammals are actually red-green color blindness. Humans are fairly unique and other primates are unique in that we can see more colors than most. Most mammals are colorblind. And I think there's this misconception that that means they see black and white, but no, they don't see black and white. This is what it sees. So I remember when I was a kid, I had a dog, and I would always throw the red ball in the green grass, and I'd be like, "What's wrong with you, dog? Why can't you find the ball?" And my dog is thinking, "Why is this moron throwing a red ball in the green grass? How am I supposed to find this thing?" So a little interspecies miscommunication going on there. And stoplights can be a little tricky too, as you can see. So here's a little link. There was a football game, I guess. It made the headlines because basically, the two teams were indistinguishable for someone who's colorblind, and it's kind of interesting to watch this kind of try and figure out the difference between who's who. So rod cells are next. Rod cells are the other type of photoreceptor. These are sensitive to dim light. They can't pick up color, but they're more sensitive to really dim light. These are located mainly in the periphery of the retina, so out in the edges. So the fovea at the center is where most of the cones are packed in, and the rods are kind of on the edges. This has another couple fun facts associated with it. First of all, a shooting star. It's actually easier to see a shooting star with your peripheral vision, because if you're looking right at a faint star or a faint shooting star, the light is hitting your cones, which see color but are not as sensitive to dim light. If you kind of shift your gaze from that star, then you're using your peripheral vision to see it, and the peripheral vision has rods, which is more sensitive to dim light, and you can actually see a star better by not looking directly at it. And so shooting stars oftentimes appear in our peripheral vision because they're very faint, and you wouldn't be able to see them if you were looking right at them because the cones couldn't detect that faint light. Rats, by the way, have almost all rods, which is why they like to be out at night. So the final thing then is just how they work. You don't need to worry about this in too much detail, but basically the way these rods and cones actually work is there's this little protein in here. It's called retinal, and when light hits it, it changes shape. If you're in organic chemistry, maybe you've heard of cis and trans. Basically, cis, this molecule kind of goes up, over, down. But when light hits it, that bond changes to what's called the trans shape, which is up, over, up. So this little change in the shape of the molecule caused by light causes it to separate from this larger purple protein, and that triggers a chain reaction. Those of you who take an AP biology, it's a cell signaling pathway that ultimately affects action potential activity, and that's actually what's happening when light hits those. That's how it causes an action potential to be fired. So don't worry too much about those details, but I thought I'd throw it in there for those who are interested. But the main idea is once those rods and cones in the back of the retina are stimulated, then the action potentials travel along the optic nerve, and then it's going to go to the brain. And hopefully you know that in the brain, it's going to be processed. It's going to go through that optic chiasma, that X-shaped structure that we looked at a little bit last unit. It's going to hit the thalamus, and then it's going to hit the occipital lobe. And interestingly, the rose that's being imaged, it's actually projected kind of backwards and upside down to the brain, and the brain kind of flips it back over, which is kind of cool. So that's it for vision. And with our next screencast, we will take a look at hearing.